Thank you guys for coming out. So I know you guys have been hearing a lot about DNA and DNA testing of the sperm and different kind of analysis. And as Dale mentioned, I'm going to tell you guys a little bit about DNA testing of the embryos prior to implantation to help improve your goal of having a healthy baby. So infertility, the struggle is real and you're not alone as you look around. 1 in 4 to 1 in 6 couples actually suffer from infertility. And it's an astronomically high number in this world. It's over 90 million couples around the world. The average age of a patient reproducing and going through IVF today is 38. And 40% of infertility is male factor related. So it's not just you as the woman patient. So first I want to start off and tell you guys a little bit about Genesis Genetics, who we are and why are we here today. Genesis Genetics is the global leading provider of pre-implantation genetic screening and pre-implantation genetic diagnosis around the world. We have 5 laboratories in North America and then also an extensive global reach, including South America, Africa, the Middle East, Asia, and in the United Kingdom. So what is PGD and PGS and why are we here? Well, what is the difference? Pre-implantation genetic diagnosis with the D is a screening of embryos prior to implantation for known genetic mutations. These are genetic mutations that you carry or you're at risk of passing on. These are hereditary diseases. A couple examples, the most common one is cystic fibrosis. 1 in 25 people are a carrier of cystic fibrosis. Other common conditions are breast cancer, Huntington's disease, sickle cell anemia, Duchenne muscular dystrophy, Tay-Sachs. Pre-implantation genetic screening, on the other hand, is a test of the screening of embryos to look for chromosome abnormalities. This is a test that's applicable to all IVF patients. And this is the screening of embryos to identify these chromosome abnormalities. And by doing so, we're able to choose which embryos are going to give you the best chance of having a successful, healthy baby. Some of the most common conditions that we can identify by doing PGS testing would be identifying Down syndrome. We can also identify the gender or sex of an embryo. We test for Edwards syndrome, Turner syndrome, different translocations, Patel syndrome, different inversions, and Klinefelters. A lot of these conditions here are chromosomal abnormalities that are compatible with live birth. Some of them result in severe mental retardation. And they involve a few chromosomes. But the bulk of them, like these chromosome abnormalities in embryos, is the reason why couples are suffering from infertility. So everybody's DNA is different. And this is why these are all customized DNA tests. Kind of go back to PGD, identifying these genetically inherited diseases in embryos. What we do is we create what we call a pedigree. And what we're doing is we're asking you some basic questions. In this example, we're looking at cystic fibrosis. Are you a carrier? You have to be an identified carrier to actually benefit from these tests. And then was it your mom or your dad did you inherit the gene from? Do you have any other affected siblings? And from there, we can create these pedigrees, and what we do is what's just called genotyping, so we can track the inheritance between different generations. And then that way, when we test your embryo samples, we can then identify which embryos are going to be healthy of a condition, which ones are going to be affected, or are they potential carriers. Now let's jump back to pre-implantation genetic screening. The screening of embryos, looking at their chromosomes. So as Dr. Allring mentioned earlier, you get 23 chromosomes from your mom, and you get 23 from your dad. From your mom, you're always getting an X, and from your dad, you can get an X or a Y. If you're female, you're getting an X. And that's XX for females, and then XY for males. And it's 50-50, completely random. But why? So here what we're looking at is the value of PGS. And the reason why is this is showing the presence of chromosome abnormalities in IVF embryos. These are shocking numbers. Even your patients, even if you're 30 and younger, 50% of the embryos you're going to make in an IVF cycle are going to have some form of chromosome abnormalities. And this increases with age. This is actually a double-edged sword, because as age increases, you're actually making less embryos. So not only are you affected by having a higher presence of these abnormalities, but you're also having less embryos to choose from. Like I said, if our average age of our patient is between 35 and 40, we're looking at roughly 50 to 70% of the embryos they make in an IVF cycle are not going to be suitable for transfer or will result in a live birth healthy outcome. So, but this is a newer technology, and this is where we've come from. This is what embryologists, the people that culture your embryos and work in the lab at your fertility clinic, used to assess embryos and choose which one to pick from to transfer. And there's some really great new technologies out here, and this was called Embryo Scope. Susie's going to talk to you this afternoon about the value of this, but allows you to monitor and watch the growth of these embryos. So before genetic testing became as popular and as validated as it is today, an embryologist would look at, and the physician would look at these embryos and choose which one, based on morphokinetics, or just based on watching the development, on which embryo they thought was the best. It's basically a beauty contest. Which embryo is the prettiest? But they're all a little different, and they grow at different rates. What I'm going to show you here, this is a picture of an embryologist, and what they do when they do pre-implantation genetic screening is they take your embryos, and this is 5 days after fertilization. And they just biopsy a little cell. These are stem cells. They send them overnight to our lab for same-day delivery. We test them, and we give your physician back a report. And in that way, your physician could decide which embryo is going to give you the best chances of having that healthy baby. So I want to show you guys what I mean by this, and why is genetic testing more of a definitive and concise answer than basically doing a transfer on morphology or the beauty contest alone. So if your average patient has 6 embryos, which one to choose from? They all look very similar, but to an embryologist and a physician, they look for certain factors: size, compacting number of cells, fragmentation within. Sorry?
I like 3 and 5.
You like 3 and 5. What about you, Doug?
Yeah, I definitely like 3.
You like 3? Okay.
Yeah.
Well, let's look at the results.
Miss America?
We'll see. Well, embryo number 1, it was the smallest one. And these are real patient results. You might not have won the beauty contest, but it is. It's chromosomally normal, it's genetically normal here. Embryo 2, well, we didn't pick embryo 2, and that's a good thing because it has the trisomy 16. So this is what our graphs look like when we read the results. And how we read these results is on the x-axis here, you have your autonomes, chromosomes 1 through 22, and then X and Y. On your y-axis, we're looking for genomic gains or losses, deviations and change. And it's a copy number variance. So you get 1 chromosome from your mom, 1 from your dad. Any additional chromosomes or loss of chromosomes is what we could reference as aneuploidy. It is going to be the leading cause of infertility. Trisomy 16 here is one of the most common aneuploidies present in embryos. So, number 3. It was pretty.
Wow.
No baby. Number 4, yeah, it has some fragmentation. It is a little bit noisier than up here, but it's a normal. These are the embryos. By choosing the ones, not only that morphologically look the best, but that also complements the genetics, is going to give you guys, as a patient, the best possible outcome. 5, good choice, Dr. Ulrich. He's 1 for 2. And number 6, this one has a trisomy 21. This embryo was transferred and it made it to live birth. You would have a baby that would be affected with Down syndrome. This is a table that helps give you it's a predictive measure. helps sets up expectations. And this is based on sampling of over 30,000 results last year alone by next generation sequencing. The latest technology being currently used in the field of pre-implantation genetic screening. And how we read this is, over here you have your patient age, and then down here, based on the total number of embryos that you have either in your cycle or in your batch of multiple cycles for testing, to help give you guys an idea of what are the chances that I'm going to have an embryo transfer. 38-year-old patient, average age 37, 38. They only have one embryo, slightly less than 50%, roughly 40%. Yes, sir.
So why is it decreasing then that 7 embryos is 100% and 8 plus is 92?
Well, it's a bias of ascertainment data. Not everybody that's 43 and has 7 embryos is going to get 100% of the time a transfer.
Are these before you send them off, or are these genetically sound?
These are genetically tested embryos.
So you have to have 6 genetically sound embryos.
They have 100%.
So these are just embryos.
Okay, so they
Okay.
haven't been tested yet.
Correct.
Okay, okay.
Yeah, correct.
Okay.
Right, yep. And to answer your question, this is based on our data of 30,000 samples last year. I think obviously as the end number goes up, it's obviously not going to be a perfect picture for every patient, but this is just the data we were able to gather from the patients that we tested in 2015. Does that answer your question? So just to kind of summarize what pre-implantation genetics screening is, it's a tool. It's a tool your physician uses to help optimize your chances of having a healthy baby. It's been shown to increase implantation and pregnancy rates across all age groups. Why? Because 50% of embryos, on average, produced in an IVF cycle are chromosomally abnormal. It helps you avoid pregnancies that are associated with chromosome abnormalities, such as Down syndrome, Edwards syndrome, Patel syndrome. Helps improve the time to pregnancy. By giving you the data and identifying which embryos are genetically normal or euploid, it helps cut down on failed transfers, spontaneous abortions, miscarriages, and just failed implantation. Promotes ESET. ESET stands for elective embryo transfer, or elective single embryo transfer. So rather than transferring 8 embryos and hoping pregnancy ensues and you have octomom, it helps you identify which embryo for single embryo transfers help improve this time to pregnancy. So these two are directly related. You can combine pre-implantation genetic screening with diagnosis. If you're at risk of passing on a genetically inherited disease, we could test for that disease and then also screen the embryos for chromosome abnormalities in the same biopsy sample. Helps reduce the risk of miscarriage, and the great thing about it, there's no optimization. So you guys can decide with your physician if this test is best, and we can test the embryos per the order. So, Dr. Allring is a big proponent of pre-implantation genetic screening. He uses it on a large proportion of his patients. And this is one of the main reasons why his success rates are so much higher than the national average. And here are 2 other clinical case studies with some other programs we work with showing similar data, but why the value of PGS testing is being applied and becoming more popular this day and age in for IVF patients. So I just would like to end with a short video. It's one of our patient education videos. It can be found on YouTube and also on our website, kind of recapping everything we just discussed. See if I can pull it up. Does anybody have any questions? I'd be happy to answer any questions. I would try to troubleshoot some technical difficulties.
I have a question.
Yes, go ahead.
Do you separate the data, like when you test the embryos, as far as male factor and female factor?
No, we do not. A lot of times in chromosome, these chromosome abnormalities is random to an extent. And to us, it's really not important whether it's male or female. It's, you know, you guys are a couple. And
I mean, I'll just answer that question. The question was, can you separate, chromosomal abnormalities from the male versus female? So if you have an abnormal embryo, where do the abnormal chromosomes come from? I think it's kind of what your question, does it come from the egg or the sperm? Well, the answer is both, often. And we estimated, because we actually looked at this several years ago with a genetics company that was out in California, and on 300 or 400 embryos, and we were seeing about a 10%, maybe 6 to 10% were related to sperm aneuploidy itself. So in other words, the abnormal chromosomes came from the sperm. The majority of the time, it's actually both. But the techniques were different back then, and the time that we did biopsies and things were a little bit different, which can affect the results. But I think the point being is that it is both. People talk about, it's all the egg problem, but in a certain percentage, it's isolated sperm problems, but in a significant percentage, it actually is both. So that's a
How do you know which donor to
great
use then?
question.
How do you know if you need a sperm or an egg donor?
That's a whole nother question. I'm not going to answer that right now. Troublemakers. The question was to use a sperm or an egg donor, and it depends on the case. Whereas the most obvious problem, and can you identify it? It's tough. It's hard sometimes. Generally, for other conditions, we often recommend egg donors first over sperm donors, mostly because of the fact that we're able to identify abnormal sperm. We can identify abnormalities in the sperm that are chromosomal in nature. So you can, as Dr. Clemente was talking about earlier, with the Embryo Selection, nano-bead technology, you can remove a significant proportion of those chromosomally abnormal sperm to have a lessen the impact that sperm defects have on the embryo in general. So you can remove at least 6 to 10%. At least in theory. We're working on trying to prove this, but some of the preliminary data suggests that at least 6 to 10% of the embryos will be automatically made better by the sperm selection process, and perhaps even more than that. It again depends upon the individual case, but it seems to be holding true. But it's always the egg problem, right?
Any other questions? Great, thank you guys.